I'm just going to talk. There will be no slides, no visual prompts, no assistance of any kind. It is your responsibility at this time of the morning to actually look at me and see my mouth move. I have no fresh data to present, and as I just said, I have no PowerPoint presentation to show. So, why would a retired investigator like me be invited to speak? I'm here, I imagine, because I was a somewhat early adopter of the current interest in interoception, though the original interest can be traced back at least as far as a Russian investigator named Ivan Sekhenov in the middle of the 1800s, and I'm sure that the Greeks knew about it too, because they know about everything. And also, I think I'm here because I was a bit of a synthesizer. As was just mentioned, I wrote that book, but that book was written more than 15 years ago. What could I offer here to this expert audience today that would be hopefully interesting and possibly useful? My talk today is a personal perspective on interoception that is speculative, theoretical in places, and first it will briefly describe why and how that book was written. In order to tell this story, I beg your indulgence, since I will be saying many things that many of you are at least somewhat familiar with already. It is admittedly self-referent in the first part and a bit far-ranging in the brief third part. And I apologize, as you can already tell, for reading some of my notes. I know people don't like people who read their lectures, and I apologize for that, but this old memory is not quite as nimble as it used to be. My title for today's talk is From Where Do Symptoms Come? It needs an explanation. As a researcher and a clinician, the trail started for me by trying to answer that question, that query, which of course we're all still doing. We should start by addressing the question, what is a symptom? While often the word symptom is used in a clinical context, and by the way, as you heard, I have always been a practicing clinician. I have had my own patients, and I have seen patients always. In the clinical context, to refer to an internal subjective manifestation of an illness, as opposed to a sign, which is an outward observable indicator. The word is also used, however, with a much broader, more general meaning. And if you want to get a really vague and general definition, follow the next one. One unabridged dictionary offers as its first definition, quote, any phenomenon or circumstance accompanying something and serving as evidence of it. That really nails it, doesn't it? It could be anything. A variety of definitions of interoceptive symptoms have been proposed, as you know. My own would involve an internal subjective experience, but not be limited to disease, and it would encompass the skin and all inside it, including both more and less accurate perceptions. I've chosen what I call a geometric dimensional heuristic. Doesn't that sound academic? For the main three sections of my presentation. I call them first the line of development, where I'm going to describe a bit about how I came to write that book, and I think there will be a bit of a lesson in it for us all. The second section will be called what I call the square of function. So we've moved from one dimension to two dimensions. And that is essentially a follow-up exposition on my 2009 paper, Visceral Brain-Body Information Transfer. And then the third section will be what I call the sphere of relevance, in which I try and give a couple of examples of how maybe the things that we care about and talk about might be of interest to the wider world. In each section, I will endeavor to answer my title question of where do symptoms come from in some way. I primarily wish to prompt some thought and discussion. Of course, not fully answer the title question. I apologize to those authors, including several who are in this room now, whose work is described but without giving specific citations. I thought that would just kind of bog us down. So the first section is what I call the line of development. I will start with an old joke that many of you might have heard before. To be an expert clinician is to know about many illnesses. However, there is only so much room in anyone's head, so as knowledge becomes broader and broader, it inevitably becomes shallower and shallower about each individual disease, until that clinician knows nothing about everything. Alternatively, an expert investigator needs to know one area in great depth and detail, learning more and more unavoidably about less and less, until that investigator knows everything about nothing. So bear that little vignette in mind. We're going to return to it a little bit as what the lesson is, if you will. I will now briefly describe my own knowledge development, which led to me being able to write the book that I wrote. And it actually led to my interest in interoception generally. In college, I studied experimental psychology, James and Lang, everybody's favorite people, conditioning and learning, attention and arousal, motivation and emotion, the work of Pavlov and his students, biofeedback, and 1 and 2 process learning theory, all of which are related to interoception and comprise the first third of the book. Let me ask for a second. How many people in the room know what the issue of 1 versus 2 process learning theory is? Does that sound familiar? Vaguely. Okay. Well, let me give a little historical note because it is of relevance. Back in the 1960s and probably earlier, there was a controversy. And the controversy was, it was claimed that only Pavlovian conditioning methods could condition autonomic responses, and only operant conditioning methods could condition so-called sort of the voluntary nervous system. There wasn't any evidence for that, so a number of investigators, most prominently a man named Neil Miller, who some of you might well know, he was, I don't know if he's still alive or not, he might be, who was a truly illustrious investigator, set out to try and find that out. And what they tried to do was to operantly condition autonomic responses. So the controversy was there 2 processes which were distinct, or was there 1 process? In other words, could both types of conditioning condition both types of responses? And there was a great deal of research done trying to answer that question, and I would suggest that to this day, we don't know the answer to that. But people started calling the whole field biofeedback, and that's where the term comes from. Biofeedback was not initially about clinical care. Biofeedback was initially about research. If you, and I will refer to Saab's paper on biomarkers, he shows this nice graph where he shows the number of publications, and there's a big biofeedback blip in the history of the publications. You're going along, minding your business, it's the late 1960s, maybe earlier, maybe a little earlier, and there's a blip in the number of research publications. That is what I would call the biofeedback blip. And then, although the clinical practice of biofeedback continued, the research went away when it became clear that nobody could answer the question. So the curve goes down, it flattens back to 0 for a while, and then it, of course, picks back up, as you know. That phenomenon, that issue, for my money, if you will, is one of the seminal questions, theoretical questions, that actually got this ball initially rolling, that stopped rolling for a while, but then picked up again. Okay. Subsequently, after college, I studied neuropharmacology, neuroanatomy, and neurophysiology, and a few other things during medical training. There was more neuroscience, which, by the way, in those days was typically called biopsychology or physiological psychology. I don't know when the word neuroscience sort of gained the coin of the realm that it has now, but when I was an undergraduate, nobody would have had any idea what that word meant. I mean, you could have parsed it out, but you wouldn't have used it because nobody was using it. And I was particularly interested in behavioral and autonomic pharmacology and endocrinology. I studied disorders of brain and biology in medical school and in residency training, particularly, as was mentioned, anxiety disorders and psychosomatic medicine. Well, almost enough about me. How did I get particularly interested in interoception? Many of you may have sort of a day when you had an idea or a thought, or in my case, my advisor essentially suggested to me part of a project of my dissertation that is really, and I literally remember sitting in his office the day he said this, that really sent my scientific career all the way along. When I was a graduate student, I wanted to see if I could use a drug state to be an unconditioned stimulus in a classical conditioning paradigm. There were other people doing it at that time. It was not a completely new idea, but it was just kind of fresh at that point, and it was very interesting. And I started that research, and it was kind of working and going along. And my dissertation advisor said to me, well, if you're going to study that stimulus, why don't you study them all? Why don't you study the drug as an unconditioned stimulus? Why don't you study the drug as a conditioned stimulus? Why don't you study the drug as a discriminative stimulus? Why don't you study the drug as a reinforcer? So that was what got me started. And now another little historical note. This research was going on from 1969 to the early 70s. Anybody guess what drug we were using? Think back. Bingo! Yes, we were studying LSD. We were using the LSD stimulus state to see if it would work in these various capacities. So basically what happened, to my own thinking, was if you can use an exogenous substance, a drug, to produce a discriminable stimulus, is it possible or even likely that endogenous substances are doing the same thing? Are we going around discriminating our humors every day? The answer probably, with one exception, is a qualified no. And I think that the reason, well, a reason, these drugs may be intrinsically non-discriminable, but things like cortisol and so on, there isn't really, there may be a cortisol stimulus if you gave somebody enough of it in a bolus, but in the time course of the way the substance is changing under normal physiological conditions, it doesn't seem to produce a robust stimulus, if any stimulus at all. But there is one exception. You all know it. You've all said, yes. You know, how many of you have said, I felt my adrenaline pumping? Come on, raise your hands, admit it, or at least thought it. Well, number one, you're able to do that. And number two, all the evidence suggests that most of the time you think it, you're wrong. The amount of adrenaline, the fluctuation in the amount of adrenaline that occurs under most physiological conditions is not sufficient, it does not appear, to produce a robust stimulus. So what's really probably going on when you say, I felt my adrenaline pumping, is things like you felt your heart beating fast, and you thought it was that adrenaline that was circulating in your blood that was doing it, but it wasn't the adrenaline that was circulating in your blood that was doing it. It was the local innervation of the nerves driving the heart. And in fact, it probably wasn't even the heart. It was probably the baroreceptors in your aorta that you were really feeling as they were being bombarded by harder and pulses of blood out of the heart. So anyway, when I started asking myself, are endogenous substances discriminable, is when I think, for me at least, interoception became a question that I cared about. Okay. Getting back very briefly to the book, one more paragraph and we'll stop talking about that. All this stuff that I learned in college, in medical school, in graduate school, in residency, et cetera, et cetera, et cetera, all of that stuff is what made it possible to write the book and what got dumped into the book. There were approximately 950 references, and I needed to know all that stuff to be able to write it. This is not the only book on the subject, but there aren't very many. But two I would recommend to you, one of which I'm sure you already are aware of, Bud Craig's book, How Do You Feel? Another book I would mention, which is probably a bit more obscure, is a book called Visceral Perception, Understanding Internal Cognition by a man named Georgie Adam. How many people are familiar with Georgie Adam? If you're an interoception researcher, you need to be. I would suggest that over the long haul, if you really want to sort of look the stretch, he's the most important person, who is, at least a year ago, 2 years ago, was still alive, because at the age of 98, he sent me an email. So I would suggest that you familiarize yourself with the stuff of Georgie Adam. He's the transition figure, I think, between all the research that was occurring in Eastern Europe and Russia, and then into, quote, the West, unquote. So there are other books, and I would certainly recommend at least those two to you. Getting back to that joke that nobody laughed at, I think the lesson to be learned, or at least the lesson I would suggest, is that for most of the people in this room who are researchers mainly, it's very easy and maybe necessary to become very, very focused in what you're doing. But for me at least, and I think it's probably true in general, it's really necessary to maintain a more broad picture if you really want to understand what you're doing and how it fits into stuff in general. So my lesson, I guess, is don't know nothing about everything or everything about nothing. Know a lot about a little, but at least a little about a lot. Okay, I will briefly complete this section by mentioning the two articles that I distributed as part of the request for articles from all the speakers. They essentially, they illustrate pharmacological approaches to studying interoceptive issues. It's been known for almost, okay, I'm good. It's been known for almost 100 years that from multiple studies that administration of adrenergically activating substances do produce symptoms referable to the sympathetic nervous system, although, as I said, really almost invariably in non-physiological concentrations. The isoproterenol paper, the exogenous administration of isoproterenol the brain's reaction to it, I think illustrates the model. It's one illustration among many of the model of using exogenous challenges to do interoceptive work. In this particular paper, we used cerebral glucose metabolism. The right insula was activated, the left somatosensory cortex, and the medial cingulate gyrus. Other investigators, some sitting over that table over there, have reported activation in the right insular cortex. And generally, the areas that were activated are consistent with interoception brain research from other techniques. So it's kind of reassuring that it looks like it was producing reasonably valid information. The other paper looked at endogenous GABA in the brains of people with panic disorder. Do most people here know what panic disorder is? It's safe, people with a lot of somatic symptoms, among other things. And we were interested in whether or not there was any GABA abnormalities in people with panic disorder. What we found in our study was that there was a decrease in GABA binding in the insula bilaterally. This followed up on a paper by Malaysia, who had reported activation. Let's see, I think it was the right insula, but not the left. There was one prior study that had seen evidence of insular change. Subsequently, there have been a number of studies that have looked at GABA in people with panic and other anxiety disorders, and they're consistent in the fact that the studies report decreases both in binding and in GABA content in various kinds of anxiety disorders. They're not consistent in the anatomical region in which they're reported. So we're kind of 1 for 2 there, but we'll see how that plays out. There's also one study in normal subjects that reported changes in insular GABA concentrations in association with interoceptive awareness. Okay, so that's kind of my end of section 1 of my paper. It reminds us that peripheral and central nervous systems activity are both places from where symptoms seem to come. Okay, now part 2. Given where we are having this conference today, it's not surprising that the main focus of this conference is the brain. But I wish to say a word for the periphery. Let us envision a basic brain-body information transfer model. Imagine a square, okay? Attend to the 4 sides of the square. We will visit each of them in turn. The top side represents the central nervous system. The bottom is whatever peripheral organ you happen to be interested in at the time. And the 2 sides are the efferent and afferent connections between the brain and the organ of interest. Basically, it is my contention that in this model of interoception, the efferent side and the afferent side are of equal importance and are essential to the whole process, as I will say. Okay. And remember that when you think about an organ that you want to study, you don't have to be stuck with just the so-called usual organs. The diaphragm is an organ that probably relates to interoception. The baroreceptors in the large blood vessels are certainly important. In fact, they may be the place where so-called cardiac interoception actually happens. And even phantom organs. There is evidence of the existence of phantom bladders, phantom ovaries. There are phantom and that people will report symptoms from these phantom organs. There's even a report single case of a phantom gallbladder. So it's not so easy to have your organs just removed. They hang around. Okay, so what I want to do is I want us to talk a little bit about the four sides of this hypothetical square. Actually, we're only going to talk about three sides. I'm going to leave the rest of the conference to have fun discussing the top of the square. Everybody else is going to be talking about the brain. As I said, I will focus on the other sides outside of the brain. The question for consideration is, what are the roles of these peripheral parts to the dynamics of interoception? It seems that a full understanding requires a resolution to this question. The first issue concerns the peripheral organ. As an organ participates in the interoceptive process, is it changed in any persistent, relevant way? As an example, returning to pharmacology, changes in tissue receptors can not only affect moment-to-moment function, but also up and down regulation of receptors can change organ function over longer time frames. Are the dynamics of interoceptive loops, and I call this square, if you will, a loop, are they influenced by such processes? Is the brain speaking to or listening to functionally the same or a different peripheral organ from second to second and day to day? That's one question. Second question, relating to the peripheral nerves. I mentioned the vagus as illustrative because it's the focus of the related research I'm going to mention, and it's certainly my favorite nerve. And I would just sort of as an etymological note, the word means wanderer. Some of you may know that. That's the translation. And it's the same root as vagabond, vagrant, and vagaries. They're all kind of things going around. Okay. So, is the vagus and are the peripheral nerves in general that are involved in this loop or these loops, are they just in quotes, passive conduits of information generated from the central nervous system or the peripheral organ under consideration? Or do these nerves play any active role in the interoceptive process? Again, I will turn to pharmacology for an example of a possibility. In the brain, at least, nerve axons and often cell bodies and dendrites have modulatory inputs, the so-called excitatory and inhibitory postsynaptic potentials, that, as you know, raise or lower the likelihood of a nerve transmitting an impulse. Could it be that modulating effects, such as receptor effects on the nerve cells, influence these visceral peripheral nerves on their way in some way? As one example of one study, administration of angiotensin inhibits vagal action on the heart, even in an in vitro preparation. It is important to realize, though, that this and other pharmacological experiments do not demonstrate conclusively if the effects observed are on the body of the nerve per se, or at the junction between the nerve and the other tissue, i.e. the peripheral organ or the central nervous system. That is it at the nerve, is it at the sides of the square, or is it the corners of the square? Okay. Another example is the observation that transmission of pain can involve either specificity coding or intensity coding. Mainly, what they're referring to there is a painful stimulus painful because more impulses are transmitted, or is a painful stimulus painful because it's being transmitted along different nerve pathways than non-painful stimuli? Again, raising the question of what's going on inside the nerves themselves. Okay. For those who be interested, Just this past week in Science, there was a large review of the neural circuits for pain that described their complexity in the periphery as well as in the central nervous system. The essential question is, or the essential issue is, are there any changes in function of the afferent and efferent nerves in question themselves that are intrinsic to the nerves, excuse me, rather than being driven by the brain and or the peripheral organ? Again, are they active conduits or are they passive conduits? The next issue that I would like to touch on briefly is, do these modules talk to each other? It would seem, these loops, if these loops really exist, hypothetically, really, if this model is valid, then there's a whole bunch of them. There's a loop for the heart, or maybe 50 loops for the heart. There's a loop for various parts of the GI tract, etc. It would seem that maybe these loops would talk to each other. They would correlate in their activities at least sometimes. It would seem that the answer should be yes, but do we have any evidence of that? 3 studies will be mentioned that will address this question. One relates to an interoceptive process interacting with a non-interoceptive sensory modality. In this study, awareness of visual stimuli synchronous with the heartbeat is diminished, that is, the awareness is diminished, compared to asynchronous activity, demonstrating an ongoing interaction between interoceptive and exteroceptive processing. The other 2 studies relate to interoception in 2 visceral organs. In one, awareness of cardiac function correlated significantly with awareness of gastric filling. In another, however, cardiac and respiratory measures of accuracy did not significantly correlate. Thus, the interoceptive loop does not function in isolation from at least some other intero and exteroceptive processes, at least sometimes. There has been interest recently in interoceptive prediction based on prior peripheral input to the brain. I do not think that the loop model is inconsistent with such a predictive function. Such brain-based predictive processing could be viewed as contributing to and determining set points for the loops, a kind of tuning, if you will, of their functions. 2 additional issues relate to this proposed model, merit a mention. It has been suggested by one investigator that there is a nervous system, an intrathoracic little brain nervous system pathway for the heart. In other words, there is a loop that never leaves the thorax, if you will, that is involved in cardiac function. If confirmed, one might consider it as a kind of subroutine to the loop itself. Interoceptive processes have been highly implicated in homeostasis. Disruption of homeostasis would be expected to produce dysfunction of loop activity, maybe a loss of tuning, if you will, and might serve as a mechanism and marker of allostasis and stress. Finally, from a developmental point of view, might there be abnormalities, ontological abnormalities of loop function? In other words, might there be genetic determinants? The answer would seem to be probably yes. If these loops exist, it would seem too good to be true that they would always work just right. So what I'm suggesting here is that where the symptoms come from needs to be thought of course, as not just in the brain or controlled by the brain, but sort of in this loop qua loop. More precisely, perhaps, is it even meaningful to localize the symptom source in one part or other of the 4 sides of the loop? Is the loop not an indivisible unit? Okay, the last section here. So far, this discussion has pretty much remained within the realm of neuroscience research. Others at this meeting will broaden the discussion to clinical relevance. Does the even larger world know of interoception? And if it does it see any relevance or care? 2 examples will demonstrate that the answer can be yes. First, this is quoted from an abstract of a published article. To reinforce existing scholarship, I examine a study exemplary in its analysis of embodied experience, Michael Fried's Menzel's Realism, Art and Embodiment in 19th Century Berlin, proposing that the author's reported empathetic experiences of Adolf Menzel's painting, Rear Courtyard and House, can be understood through concepts of sensory motor imaging, hypnosis, and interoception. Embodied experience, sensory motor imaging, interoception. This article, entitled Embodying Art, the Spectre and Inner Body, appeared in a journal called Poetics Today in 2010. So it's out there with the poets. Secondly, and this is my last example, surely you have thought or said to yourself or heard someone else in that larger world say, I could have been born blank, and complete the sentence. An understanding of the role of interoception in your own very specific body in forming and defining yourself would seem to, quote, prove that the answer is, follow this now, no, the you which is you could not possibly have been born or be any you other than you. Did you get that? Write that down. Not a chestnut tree, not an earthworm, not an eagle, not even an identical twin. Nothing more or less different than your own Jamesian I. In other words, no, you could not possibly have been born anything other than you, because then it wouldn't be you. It wouldn't be I, it wouldn't be you. Obviously, this issue fits well into the overall interest in interoception with evidence for cortical midline structures and the self. This is just one possible example of how a comprehension of interoception could inform general thinking. In other words, might not everyone care from where do symptoms come, at least somewhat, sometime? So, I became interested in large part because I wish to find those places from where symptoms do come. Thank you for your attention. I hope my comments have been provocative or at least entertaining, and I think we have some time for discussion. Thank you.